Welcome, everyone, to this week's edition of the Extracellular Vesicle Club. This week, we have a collaboration with the Extracellular Vesicles in Nervous Systems Special Interest Group, or SIG. This is an ISEV group that was established more than a year ago, has held its own meeting, has a lot of different activities. We will put a link to their activities in the chat box. And before I turn it over to EViNS, though, I just wanted to remind everybody about the currently ongoing period for late breaker submission for the ISEV 2025 meeting. This is a meeting that will be held in April of this year in Vienna, Austria, and you have the opportunity for the next few weeks to submit a late breaking abstract and to present your best work at this meeting. So please take advantage of that. I will also put a link in the chat box for that late breaker. So now I'd like to introduce Professor Christian Neri, who is one of the leaders of EViNS. Christian, thank you so much for being here today and for organizing this event and moderating for us. I'd like to hand the microphone over to you.
Thanks a lot, Ken. So welcome to everyone. Indeed, this is the second edition of the EViNS sponsor EV Club, and I will moderate this EV Club because it's about Huntington disease. But remember that, in fact, this EV Club series is organized by Tsuneya Ikezu, who is a colleague of EViNS, and also by Stefano Pluchino. So it's a very important activity of EViNS. And as a reminder, for those who can't follow these talks live, the archive videos are available on the webpage of EViNS or on the YouTube page of the EV Club. So today we have the pleasure to host Carla Lopes, who is running research at the University of Coimbra in Portugal. And she's going to talk about basically the effect of EVs secreted by fibroblasts, I believe, on Huntington's disease neurons and other aspects. I'm sure that you're going to enjoy this talk. And I will give the floor to Carla for the talk. If you have questions, remember to put the questions into the chat box, and then we can go through these questions at the end of the talk. Thank you so much, Carla, for being with us today.
Thank you so much, Christian, for the kind words, and Ken. So I'll share my presentation. Good afternoon to you all. So as Christian said, my name is Carla. I work at the University of Coimbra in the Multidisciplinary Institute of Aging, and I'm going to present you my work that was published in Theranostic in 2023. And I'm going to try to convince you that extracellular vesicles can improve the GABAergic transmission in the median spine neurons in Huntington's disease. So the disease was first described in the 19th century by George Huntington. So he observed several families that have certain phenotypes. He observed that this kind of disease was running in the family, so it has an inheritance pattern. Usually the symptoms was starting in adulthood. There was a tendency to progress the disease severity and to death in several years. Also, these patients have a tendency to suicide. You also observe that they have these spasmodic movements of some muscles, face, arms, which we now call the Huntington's chorea. So nowadays it's more clear what this disease is, the causes of this disease. So we know now that it's an autosomal dominant disorder, neuropsychiatric disorder. Although the prevalence is relatively low, there's a huge burden, economical and social burden, particularly for the family, since the symptoms start in middle age, and the patients die 15 to 20 years late. So the classical description of this disease emphasizes that HD has a striatal degeneration. Nowadays, we know that it's not it's not the striatal that it's all involved. We know that the cortical is also involved, is also one of the hallmarks of the disease. This disease is caused by CHG expansion in the chromosome 4 that results in abnormal expansion of a protein, Huntington, that when it has over 36 glutamines, it has abnormal function and results in this disease. We thought it always been thought that this was a familial disease, but in the last years, it has been shown that there are de novo cases that arise from this dynamic instability, or this CHG tract is highly dynamic and can originate new cases. So a lot of studies have come up, multi-century studies have come up in the last years that try to identify some biomarkers for this disease. So in this particular study, they try to identify some imaging biomarkers. So usually the diagnosis of the disease is when the motor symptoms appear. But what these studies have shown is that before the motor impairment or the motor abnormalities, the chorea, it's a decrease in the striatal volume, in the cortical volume is already present. So, and no cure or neuroprotective treatment has appeared so far. So how the vesicles, these extracellular vesicles come up here? We know that exosomes could play a neuroprotective effect or a neurotoxic role in the central nervous system pathological process. Several studies have shown that exosomes are involved in neurogenesis, synaptic function, neuronal plasticity, and neuroinflammation. We also know that EVs are key players in cell-to-cell communication. They are released by all the cells and in the central nervous system. So we know that neurons release EVs in response to calcium flux, to potassium depolarization, to glutamate activity, induced activity. And we know that there's communication, bidirectional communication between the progenitors, the neural progenitors, microglia. And very important, some studies have shown that these support cells, astrocytes, oligodendrites, they release EVs that are enriched in trophic factors, but enzymes that help to support they are neuroprotectives and help to support the survival of the neurons. So also novel therapies have arisen, particularly in the last 2 decades. There was a huge investment in cell therapy, particularly with progenitors, to try to revert this degeneration, although the results were not that satisfactory. So another approach that is being taken in the last years is this EV therapy that doesn't use cells directly, but it presents a cell-free option that reduce the risks involved in these cell-based therapies, but leverage the advantage of the stem cell secretome. So taking this in consideration, in this work, we aim to observe if EVs isolated from human fibroblasts could revert alterations observed in these median spine neurons that were derived from or differentiated from the HD iPS. So in this study, in a collaboration with the local hospital, the university hospital, we performed biopsy. They performed biopsy in nine patients, 3 controls, 3 pre-symptomatic without a motor phenotype, and 3 late stage that already have the motor phenotype. And we reprogram them and differentiate to optimize protocol into GABAergic neurons, which are the neurons more affected in the striatum. So we assess this differentiation at different time points until we have full mature neurons at day 80. So these cell lines, the 9 cell lines that we have the controls, and we have one with the highest number of CHGs, the 52, that was still pre-symptomatic, and the other ones, the lowest one was 42. From these 9 cell lines, for the study, we use 2 that we reprogram into iPS and differentiating to progenitors and neurons. So we first characterized the iPS by examining the pluripotent markers. So as you can appreciate here, they expressed NANOG and OCT4. And the first step was to see if we could obtain the neurostem cell markers. So they expressed nestin, SOX2. We continued the differentiation and analyze at 45 and 60 days. We first assessed the expression of GABAergic markers, the glutamate carboxylase, that it's a marker for the GABAergic neurons. And we also assessed DARP32 immunoreactivity because it's a marker for median spine neurons at day 45. We can see that in this graph that the cells differentiate at the same pace. We assess again these same markers at day 60, and very surprisingly, these cells seem to result in less efficiently. In median spine neurons, the yield of median spine neurons was lower in the HD neurons. We also assessed the markers for neuronal markers, MAP2 and beta-3 tubulin. We can see that at day 60, the HD neurons have more immunoreactivity for beta-3 tubulin than MAP2. So it seems that these neurons are in a more immature stage than the controls. We know several studies have shown that the expression of mutant Huntington can cause the delay in the striatal neuroprogenitor development, and it results in deficits in changes in the striatal circuits. So at day 80, it was our full the end of our differentiation protocol. We assessed the DARP32 presence as previously, but also the SMID32, which is a marker it's an axonal marker. And this protocol, although it was to obtain an enrichment in GABAergic neurons, we assess if there's some excitatory neurons. So we didn't find much for that express VGLUT, so it was mostly GABAergic neurons that we obtained. So another step was to evaluate the inhibitory synaptic density by the colocalization of the presynaptic marker synaptophysin with the postsynaptic protein geoffrin. And you can appreciate here, the HD neurons have a tendency to a decrease in these synaptic density when compared to this control. To fully assess the maturation of these cultures, we evaluated the electrophysiological properties by the capacity to generate action potential. So both cultures generate either repetitive or single spikes, indicating similar levels of maturation. What we observe further is that the HD neurons have a lower resting potential, meaning that they are more depolarized. But the threshold to generate this action potential was similar between the lines. What we did found is that these HD neurons seem to be more to have an increased excitability, since they require less currents to evoke this action potential. So next, we evaluated by a protocol of steps of voltage the voltage gates sodium and potassium channels. We didn't find much alteration for the potassium channels. We found that neurons have a tendency for larger maximum current densities for the sodium, probably due to difference in the membrane levels of these sodium channels. So since this was GABAergic neurons, we assess the GABAergic receptor mediated currents. So we apply the fast bath application of GABA at 100 micromolar pulses to evaluate these GABAergic currents. And surprisingly, we observed that there was a huge decrease in the whole cell currents of GABAergic receptors. So next, since we have to use the EVs from the fibroblasts, we first start by characterizing them. We isolate EVs from the medium of the cells. We characterize them for size, for shape, by using TEM, by using NTA. They have around 100 nanometers, which fits the characterization of extracellular vesicles. They are also positive for flotilene and CD63, negative for calnexate. So we properly isolate these EVs. Since several studies have shown that these EVs can carry mutant Huntington, so we next evaluated if it was present in the EVs isolated from the controls and the HD fibroblasts. So the wild type form of the Huntington was present in the three cell lines, the EV secreted by the 3 cell lines, but the mutant form was present. So we could identify fragments of the mutant Huntington in the EVs that was secreted by the HD, the presymptomatic and the symptomatic, obtained from the presymptomatic and symptomatic patients. So it seems that these EVs carry and can propagate the mutant form of the protein. So next, we come up with a protocol to incubate the neurons and to evaluate the electrophysiological property as previous. So since the 80 days was the full maturation, we decide to start applying the EVs every other day from 5 days before the 80. So it's starting at 75 every other day until the 80, which a known concentration of EVs. As a proof of concept to guarantee that the neurons could uptake these EVs, we overexpress in fibroblasts in one line of fibroblasts CD63 tagged with GFP. We isolated the EVs by ultracentrifugation and did this 3D reconstruction. And this, as you can appreciate here, the EVs are in the neurons. So after we proceed as previously, we evaluated the GABAergic current density in the HD cultures that were treated with EVs. And very surprisingly, we have a normalization. So incubating these cultures with EVs, with control EVs, we have a normalization of these GABA receptors currents. And the opposite effect occur in the HD, in the control neurons. When we incubated it with the EVs from HD cells, we have a phenotype that is very similar to the HD neurons. So this was very surprising. So next we try to evaluate what could be mediating this. So we evaluate the miniature inhibitory postsynaptic currents, the amplitude, that we didn't see much difference, but we did see difference in the frequency. This is the EViNS interval. The frequencies is the opposite. So we can see there's an increase in the frequency of these minis. So from this, we can assume that the EVs are increasing the number of synapse contacts, but not the number of receptors, of GABA receptors in these cultures. To further confirm this aspect, we evaluated the GABAergic synaptic density by performing colocalization as previously from synaptophysin and trypsin in these cultures treated with EVs. And it increased the number of the synapse contacts. The inhibitory synapse density was increased when we treat these HD cultures, these HD neurons with the EVs. So we know that EVs can they carry a different cargo, but they are involved in, it has been described that they are involved in neuroprotection by transporting microRNAs. They are important in cell-to-cell communication through the transport of biomolecules. So we decide to look for the microRNAs inside these EVs. So we analyzed, we did high throughput screening sequencing of the microRNAs within EVs. We found that there were around 393 microRNAs in all the EVs. We took the 100 most abundant microRNAs in these EVs, and we found that two different clusters that that could aggregate to different groups, one for the pre-symptomatic and the other for control. Very interesting. We also saw that that there were 227 microRNAs common to all the groups, but some of the microRNAs were exclusively detected in a specific cell-derived EVs, confirming the selective incorporation of these microRNAs into EVs. Unfortunately, probably due to the low number of samples for each group, we could not find differently expressed microRNAs. So we did a different approach. We look for the changes between the groups, between the control, the pre-symptomatic and the control, the symptomatic, the control, and the symptomatic and the pre-symptomatic. And the most changes as expected was between the symptomatic group and the control, and the least was between the late stage and the pre-symptomatic group. Among the most enriched microRNAs that were common to all the groups, we found these three: the 576 and the 1260 family, that were both overexpressed or upregulated mostly in HD. So you use the MIR DB database to predict the target genes of these microRNAs. And we focus our analysis on the targets that were involved, the genes that were involved in neuronal and synaptic components. So we found that some of these, we found an enrichment for targets in the synaptic genes that are involved in neuronal cell body, dendritic, axon, presynaptic membrane, neuroprojections, and axons. So since this was a general database, we decided to proceed by using a specific database that was focused on synaptic genes, the SynGO, the database is only for genes involved in the synapses. So we found that from our microRNA targets, the up and down regulated, we found that around 10% of the genes that were differently expressed between the cell lines were mapped to these SynGO annotated genes. So this is interesting because some of the microRNAs could really target the genes that are involved in synapses. So since we used only two EVs obtained from two cell lines, one control and one HD, to perform this reversion of the phenotype that we observe, we decide to focus on the context of these EVs. So first, most of the EVs, most of the microRNAs are shared between these different EVs, so between the control and the HD EVs. And as previously, we know that there are some specific microRNAs for each of the condition. So since we just have two groups, we decide to go for a different analysis. So we look for the number of reads, and we look for the reads that were at least to change by 2-fold between the controls and the HD. So we're able to identify 7 microRNAs that were at least double upregulated in HD compared to the controls, and 7 that are downregulated. So to better understand if those genes were involved in synaptic biology, we applied the SynGO analysis to the target genes as we did previously. So we observed that the downregulated targets of the microRNAs that were downregulated, they have targets in the synapses that they were involved in the synaptic organization, in transport, metabolism, signaling at the pre and post synaptic compartments. For the upregulated GO enrichment, we obtained similar results, but they were mostly involved in the synaptic organization, synaptic vesicle cycling, signaling, among other functions. So the next, to further see, so this is a general list. If you go to SynGO, it's a general list that has genes involved in synapses. So we want to go further. We want to see the list to check which are the genes involved in the inhibitory synapses. So Dr. Matthias kindly provided us a database only with genes involved, validated genes involved in these inhibitory synapses, and we performed the SynGO, through the SynGO, we performed the same analysis to see which are the list of genes that are targeted by these microRNAs, particularly in the inhibitory synapses. So we observed that at least for the, we found potentially target genes that were by upregulated microRNAs in HD, and they were mostly involved in postsynaptic organization, regulation of translation at synapses, synaptic transmission, and regulation of synaptic organization. So it seems that the microRNAs that are carried by these EVs, they can target the proteins at the synapse. They can modulate its presence or its at the synapses. So as a wrap up of this work, we've shown that the differentiation of the iPS into neurons results in the decrease in the median spine neurons differentiation. We know that there's a decrease in the synaptic functional maturation with the decrease in the whole cell GABAergic receptor-mediated current densities. We also know that EVs can carry the mutant form of the protein, and very interesting, when we treat these striatal-like neurons, we can reverse the phenotype abnormalities, particularly the decrease in these GABAergic currents. And also we found that these EVs are enriched in microRNAs that have targets at the synapses, and they are related to inhibitory synapse functions. So probably this is just a follow-up question. Can these microRNAs that are in these EVs have a role in functional development impaired that we see in patients with HD? So I want to thank you all for being present to listen, and also the collaborators that participate in this study, Ricardo, Paulo, Conceição. We are here today to answer, to help answer the questions, our collaborators, and also Margarida and Rita for helping with the work. And I'm happy to answer your questions.
So thank you very much, Carla. I guess that there will be questions.
Sure. Okay.
I don't see any question in the chat, but perhaps someone would like to ask a question. If so, you could raise your hands and can unmute. Okay, so there is a question from John Bisler, which is about is the magnitude of EV suppression similar to vigabatrin?
To what?
To vigabatrin. There's a drug that suppresses GABA.
I don't know. I cannot answer that. What we know is that you're talking when we use the HD EVs, the suppression. I don't know, the levels that we obtained were similar to the HD cultures. So about the drug, I don't know. I never tried, but it was amazing, the results, the decrease in these GABA currents.
That's really cool. I just was trying to understand sort of magnitude, is why I asked the question. So
I don't have the. I never tried that drug, so I don't have the idea. But I've tried to be and can
answer the goal. Yeah. Thank you so much.
Thank you.
So we have some other questions lining up. So there is a question from Seiko Ikezu, which is about whether you tested the effect of control EVs on control neurons, and also, which is my question, whether you did the same for HD EVs on
to see if it could enhance. No, it didn't. That will be a nice follow-up. At the time, we're just so stunned with the results, because we're very surprised. We're not expecting that the reverse of these, at least the GABA currents, was so amazing. But we didn't. That could be a nice control also. Unfortunately, we didn't.
Another question also, did you try to mimic the studies using any of the microRNA activators or inhibitors?
Yes, that will be also a nice follow-up for the project, to see if we can isolate a microRNA that really has a target, and see if it's some of the proteins are not upregulated. Unfortunately, we didn't follow up that. We had other projects, and we just stopped with these. But I think it's the following step to really validate some of the microRNAs and see if they can be used, can be useful in this disease.
Another question from Ines Caldera Bras, saying, great work. Did you look for HTT transcript in the EVs? Actually, this is an interesting question, because I had also some questions about the detection of Huntington in your EV extracts. But first, I mean, how about HTT transcripts?
No, we look for them. We did immunogold, and since we found it, we didn't went for the transcripts, because other studies have already shown in 2016 and before that the transcripts were there. So we just want to, because some of these effects, at least when we use the HDVs, I think can be the cause can be the mutant Huntington, that it's already been shown to impair these neuroprogenitors' development. And the patients, they have decreased brain volumes, the cortical, the striatum. So I think the mutant Huntington has a role here, particularly in the HDVs. In the controls, I don't know. MicroRNAs was our what we searched for, but many other bioactive molecules could be also regulating this.
So in the conclusion, you say that basically your EVs, the EVs that you have isolated in this study, they contain a mutant Huntington. But since this EV population is probably highly heterogeneous in terms of size, in particular in terms of origin, what do you think of this heterogeneity? I mean, so that's one question. Another question is whether you are sure that using the 1C2 antibody, the signal that you see in electronic microscopy experiments is really, about mutant Huntington, or whether it could be that there is some sort of cross-reactivity with some other motifs that would be in these EVs and that won't be mutant Huntington. I'd just like to have your feedback on that, particularly as to the heterogeneity, and whether you have looked since your data were published, whether you looked at EV subtypes, whether you looked at
In this culture, they are more or less homogeneous. So here you can just have astrocytes, and we have less than 20% of the astrocytes, so they are mostly enriched in GABAergic neurons. We don't have much glutamatergic neurons, so I think most of the EVs that are released are from the neurons, because that's what we have in the cultures.
But these are EVs from fibroblasts, right?
These ones are from fibroblasts, yeah. So it's
right.
it's from skin fibroblasts, so I'll say they are quite homogeneous. There's not a lot of cells. There's not other cells on culture. About the cross-reactivity with these 1C2 antibody, one thing that we try other antibodies, but we pursue with this one because we didn't find any in the control EVs. So in the control EVs, we try several different experiments, and we never find it. So that's why we thought that could be specific for these for the EVs that were released by the HD, and that's why we didn't look further. Actually, it was not the aim of the work to look for the transcripts. We could further explore that. That's true, but it was already published, and it was already established that they were carrying. So we didn't follow that path.
So there is also a question again from Seiko Ikezu, which is about, do you believe that the effect of control EVs are specific to GABAergic neurons? And is it supposed to deliver therapeutic effect on excitatory neurons as well? I think one question also beyond that is, I mean, what is your prospective view on the next step of your study? I mean, are you proposing that EVs isolated from wild-type fibroblast could have some therapeutic potential against Huntington disease? I mean, what is the,
Yeah. This I know that's not a lot of studies with fibroblasts, with skin fibroblasts, at least with this approach. There's a lot of mesenchymal stem cells, and they also have very good results in different diseases. With the fibroblasts was a bit surprising. So I think the contact EVs could also benefit some of the excitatory synapses. We didn't try that because our protocol was aiming for these inhibitory synapses. But I think that the problem is that the contact in EVs is so heterogeneous that it's hard to isolate one target to say that is from that the reversal that we see or the benefit that we see is from this or from the microRNAs or from the trophic factors or from I think it's difficult to isolate just one element, one component. So in this case, we target for the microRNAs. The following steps should be validated, see if the genes are really upregulated or downregulated, and that can have an effect. Yeah, but unfortunately,
I was also wondering about how many EVs you incubated with how many cells.
A lot. That's why you use fibroblasts.
Because you have some sort of a protective effect, or I would say sort of a normalization of the signal. And the question is, since they appear to contain mutant Huntington, then is there any toxicity related to the presence of a mutant Huntington in your EV extracts, that could, be involved in this?
The results with the
yeah, so I mean, what would be and that relates to a question that is in the chat box, which is about the cargo content from fibroblast to induce this specific effect that you see on GABAergic neurons. The question is whether you think that it could be an unspecific effect on membrane stimulating or other release of other vesicles in the cultures, in the neuronal culture, So basically, do you think the effect is direct or could be,
that's a good question. I don't know if I can answer that. If that can have a positive loop effect, I don't know. It's hard about the quantity. That's the quantity of EVs. That's why you use fibroblasts, because it's easier to. Because in the beginning we are thinking about using the neuronal EVs, so isolated from the neurons and do these experiments. The problem is that the yield is very low, so you use around 20 million EVs for each of the incubation, which is a lot. If that can have a toxicity effect for the EVs isolated from the HD patients, perhaps, yeah, that's a question. We did some titration. We look in the literature if there was some reference for these positive effects. We didn't find much, though we did a small titration to see which was the amount that we can use, and we got some results with this protocol, the 5 days to the 20 millions in each of the days. If it's the toxicity by the amount of mutant Huntington, probably. But I think the most important results here is also the reversion, it's the control EVs. So what is being secreted by the fibroblasts that could really rescue this impairment in this in the development of the synapse. Yeah, I think that's the other one was just a proof of concept that there's something, maybe there are different factors that are regulating. Probably the mutant Huntington is regulating the depression on the GABAergic, and in the control EVs, there are other factors that are improving these the synaptic function in these GABAergic neurons. So probably there are different mechanisms of action. And if they can induce positive for I don't know, because in the HD cultures they are homogeneous. You don't have very heterogeneities. You have some astrocytes and mostly GABAergic neurons and some glutamatergics, but they are very residual.
Another question from Eken Anakor. Do you reckon that the mutant Huntington are only resident in the EVs that were isolated, or would they, for example, be present in other types of extracellular vesicle? That relates to the former discussion we had.
If the other types of vesicles could carry the mutant form of the Huntington, yes. In this case, we have its small vesicles because we filtrate before adding it to the cultures. You use a 200 nanometers filter, so most of the large EVs are eliminated. And when we extract we maintain we isolate the EVs, we freeze them at -80, and then whenever we want to use them, we filtrate and we put them in the neurons. So I'm sure that it's small EVs. I don't know if that was the question, but the large EVs, for sure, they will carry also the protein.
I believe it would be interesting to really dig in, the type of EVs that carry mutant Huntington by looking at using other antibodies against Huntington, like sort of ELISA, sandwich ELISA, to make the difference between wild type and mutant Huntington. That could be one interesting experiment. Also, I think it would be interesting to know whether the EVs that contain mutant Huntington, whether they are larger vesicles, like it could be exopheres, these are very large, of course, but talking about, ectosomes or microvesicles, whether those would contain mutant Huntington, whereas some other EV subtypes would not.
Yeah, I agree. Probably these ones will have just fragments or the transcripts, mostly the transcripts, because they are very small, but the large ones could carry the protein itself in the full length, probably, depending on the size. Yeah, the impacts of those EVs on if you carry the transcripts or if you carry the protein. I think it will be also interesting to see which is the impact of in this kind of phenotype.
I was also wondering, I think that we reviewed all the questions that are in the chat box, so maybe we can go with some additional discussion. There is one mechanism that is emerging in the field, of Huntington disease research, which is somatic expansion. And somatic expansion is about larger KEG repeats compared to what is inherited at birth. And a major question in the field today is, what is the threshold for toxicity? Are we talking about 55 repeats, or do we need 80 repeats to disrupt a specific mechanism? Or do we need to go up to 150 repeats, in the adult brain, in the adult medium spiny neurons? So I was wondering whether you also tested, lines, human MSNs that carry larger KEG repeat sizes.
No, the juvenile, more the juvenile forms, or the, you mean?
Well, something that would basically, reflect the new mechanism that is being
the
which is
toxicity
published today by Nat Hines and very soon by Steve McCarroll about somatic expansion, like KEG repeats that are expanding in size in the course of adult life, specifically in the medium spiny neuron of the striatum.
No,
and probably also in cortex, by the way.
Yeah, that's quite interesting. This dynamic instability of this CHE. I think it's I really find it interesting because as you said, why the striatum is more affected. How can it impact this specific brain area? But we have the patients that we from the hospitals that we collect these biopsies were all within this range, so we didn't.
The question behind that is whether you think that the phenotype that you are looking at, which is the excitability of the medium spiny neurons, whether it's due to a loss of normal Huntington or whether it's due to a gain of toxic properties by mutant Huntington.
Yeah, I think it's the gain of the dominant negative effect. Yeah, because I did some studies previously also with kinesins and more related with the transport, and it's I think it's the negative effect. So the mutant form can really impair these mechanisms by doing these negative actions. So the loss of wild type can have some impact probably in neurodevelopment. It's quite important during the neurodevelopment, but the mutant form can have this negative effect and impair all of this process. So I think depends on the stage of the disease. In this case, we're trying to evaluate the neurodevelopment. So probably the lower amount of wild type Huntington that we have could also impair these progenitors' development, these GABA progenitors' development.
Mm-hmm. Okay. All right. So perhaps there are some additional questions from the participants. Anyone would like to ask a question? Okay, I don't see anyone raising hands. So I guess that thank you so much, Carla, for sharing this data and sharing your thoughts about this very interesting topic. And thank you to all the participants for being here today. I think that we are going to stop here and just like to call everyone to participate into the next EViNS sponsored EV Club that will happen soon, and to stay tuned on the activities of EViNS. There will be many news coming up soon for the activities of EViNS in 2025-2026. And I also like to call everyone to, if you are there, to attend the General Assembly and the social event of EViNS during the annual meeting of ISEV in Vienna. Thank you so much.